Okay, so that brings me to the next award winner, which is Liang, and he told me I could call him Liang because that was easier. Liang obtained his PhD from Bar Ilan University in Israel, and he joined IONIS in 2009. He is currently the executive director of Core Antisense Research, and his focus is on understanding the mechanics of cellular AON uptake, intracellular trafficking, factors affecting antisense oligonucleotide activity and safety, and new mechanisms to use antisense oligos to regulate gene expression. So I think everyone in this room can benefit from his work. We got a lot of good paper nominations this year, but the jury agreed that this paper probably has the most long-term impact on the field. So congratulations, Liang, and of course congratulations to everyone in IONIS who contributed to the paper. The floor is yours.
Thank you. Thank you for your nice introduction. Can you hear me? Yeah. Okay, thanks. So it's a great honor to be here. To be invited to present our work on behalf of all the authors from IONIS, the Party Wen from my group, and Mike Migawe from the medicinal chemistry group led by Puneet. And specifically want to thank Stan for his support and guidance over the years. We try to understand how ASOs work in insect cells, so how we can make a better drug for the patients with improved potency and safety. For example, like any drug, the ASOs may also have some toxic effects for certain sequences or certain designs. So for the potential mechanisms, there are several potential mechanisms have been proposed. For example, the hybridization-dependent and hybridization-independent mechanisms. As ASOs are designed to base pair with RNA targets, so of course it can affect RNA function, processing, or trigger RSH cleavage based on the design of the ASOs. Today I will focus mainly on the ASOs that act through the RSH mechanism. So in this case, ASO-based pair with the RNA, it can trigger RSH cleavage for both on-site and occasionally for off-target RNAs if there are the decent-based pairing. So if the encoded gene has some critical function, it can cause toxic effects. So this model was supported by the recent finding by Sebastian from our company, which was published several years ago. It showed ASO toxicity is RSH1 dependent. However, there are some observations cannot be easily explained by this mechanism. So for example, through bioinformatics analysis, it has shown there is no strong correlation between the off-target size and ASO toxicity. On the other hand, the toxic ASOs have no species specificity. So this is important, especially when you think about the potential off-target size. In many cases, it's predicted to locate in the intron region, which are poorly conserved. So these observations led us to think about some extra mechanism to explain these observations. These go to the ASO protein interactions. We know that the ASOs, especially for the chemically modified ASOs, can bind proteins. The ASO protein interaction can affect the protein conformation, for example, and also can affect the protein binding to its partners. So this may lead to alter the protein function, localization, or stability. So if the proteins are affected, they have some critical function. Of course, it can also cause some toxic effects. So over the last 7-8 years, we have spent a lot of efforts to understand the ASO protein interactions. So what proteins the ASOs bind, how they interact, what are the consequences. Now we know that many factors from the ASO side, they can affect protein binding. As it is well known, phosphodiester, phosphosylate backbone is required for protein binding in a position and number-dependent manner. 2' modification have very important role in protein binding. With more hydrophobic 2 modifications, have stronger protein binding. In addition, it's also sequence dependent. The different sequence can have 1,000-fold difference for the binding affinity. And one thing I want to point out is the azo-protein interaction shows some polarity effects. The proteins tend to bind to 5' wing with more hydrophobic modification, though together with around 6 nucleotides in the DNA gap. At the protein site, not surprisingly, many azo binding proteins are DNA RNA binding proteins, and also there are some chaperone proteins as well as other proteins. So we have found that the azo protein interaction can affect azo activity through different mechanisms, and also it can affect the protein fate based on the binding affinity. So this may affect azo safety. So based on that, we reasoned if the azo protein binding contributes to toxicity, it should act faster, because after azo enters the cells, they could immediately bind to the proteins. In this case, a more detailed kinetic study should help us to better understand the mechanism of azo toxicity. With better understanding of the mechanism, we might be find a way to modulate the azo performance. So before the kinetic study, we first want to identify some toxic indicators so we can follow that in the time course studies. As we can see here, first we found the most of the toxic azos, here I focus on hepatotoxic azos, actually trigger cell death through the apoptotic pathway, as I showed the example here. For the 5 toxic azos and not the 5 safe azos, it can cause elevation of ALT, AST, and also cause increased mRNA levels for some genes that are p53 targets. In addition, the toxic azo, but not the safe azo, cause caspase activation, and also it causes PARP cleavage, to suggesting apoptotic cell death. Another interesting thing is we found that toxic azos can cause protein mislocalization is part of perispeckle proteins from the nuclear plasm to the nuclear line. So you may know that perispeckles are nucleoplasmic RNP granules. It contains a long encoding RNA called NIT1 and dozens of proteins. It's part of p54NRB, another name is NONO, or PSF. As you can see here, that the PSF protein localized in distinct fossa in the nuclear plasm. When we transfect 2 different toxic azos, we found these azos tend to localize to the nuclear line, and also it caused p54 protein mislocalization to the nuclear line from the nuclear plasm. But this was not observed with 2 safe azos, whether it's localized to the nuclear line or not for the azos. We have observed more than 100 pairs of azos. In all cases, almost toxic azo cause protein mislocalization to the nuclear line, but not the safe azos. So similar observation was also found with the live cells where we use the GLP-tagged PSL protein. 30 minutes after electroporation, both the azo and the protein localized to the nuclear line. In addition, the toxic azo transfection also causes rapid protein degradation. 4 hours after transfection, both PSF and p54 we look at already dramatically degraded, and followed by PARP cleavage. So using these markers, including the protein mislocalization, degradation, and increased level of p21 or caspase, we look at in more detail for the toxic events, both in vitro and in vivo. In vitro, after transfection, within 30 minutes, the oligos localize to the nuclear line and immediately followed by protein mislocalization, which occurs within 30 minutes to 1 hour. And after that, we start to see the caspase or the p21 upregulation around 1 hour to 4 hours. And at this time, we start to see a decent target RNA reduction. 4 hours later, we can see the caspase activation, PARP cleavage, as well as the protein degradation at this time. After 8 hours, we can see severe cell death. Similarly, we also look at the kinetics in vivo, and after sub-Q injection, around 12 hours, we start to see increase the level of P21, suggesting activation of p53 pathway. At 16 to 24 hours, we start to see the protein mislocalization, nucleus dries, and also we can observe almost maximum target reduction. And later, after 24 hours, we can see the caspase activation as well as ALT activation, which occurs mostly at 48 hours and later. So also the kinetic study suggests the protein mislocalization actually is the first event we observed during this process. Because all these parazygotic proteins we previously found, they are oligobinding proteins. This promoted us to think whether protein binding contributes to the toxic event we observed. So first, we look at the protein binding profile using the affinity selection assays that we used. Oligobinding proteins were selected using a built-in labeled oligo and then eluted by competition with ASOs with either different sequence or different designs. So we can compare the protein either by silver staining or by Western analysis. As we can see here, the toxic ASO as compared with a safe ASO, they bind much more proteins and more tightly. It doesn't matter which capital oligo we use. In addition, the toxic ASO and not the safe ASO can form a distinct ASO protein complex, as we found using a EMESA assay. A strong correlation between protein binding and toxicity was also found through a polarity studies, as I mentioned earlier. The many proteins tend to bind 5' polar, and in this case, we changed the 2' modification for different wings, and so we can achieve different protein binding profile. For the ASOs that showed weaker protein binding, they appear to be safer as well. So if that's the case, the next question is, so can we modulate the protein binding using chemical modifications so we can improve ASO performance? Because we know most proteins, they bind to the 5' portion, including some gap DNA sequence. So we introduced different 2' modifications at gap position 1, 2, 3, and 4, and then we look at the effects on ASO performance. As we can see here, when we introduce 2' O-methyl at gap position 2, we can dramatically reduce toxicity if we look at the ALT levels with the comparable activity as the parental ASO. So suggests that when we introduce a single 2' O-methyl at gap position 2, we can dramatically reduce toxicity with no or little effects on the ASO activities. So this was further confirmed by a full positional work. In this case, we introduced 2' O-methyl at different positions in the gap from 1 to 10. Thanks Holly for introducing the design of the ASOs. And only at position 2, we have dramatic reduction for the toxicities, and also have a dramatic reduction for protein binding at position 2, and also cause the protein relocalization to the nuclear plasm, and reduce the toxicity was also confirmed in vivo in a more detailed dose response studies. So improvement by O-methyl at gap position 2 was also confirmed using a different toxic sequence. In this case, only at position 2, as compared at position 1 or 3, it shows the best benefit. It also works for different design. At Ionis, for the Gene 2 design, it's the 5-10-5 uniform PS-Mo ASO. In this case, O-methyl at position 2, but not at other positions, also dramatically reduce the toxicity. So suggesting that O-methyl at position 2, a simple modification, can dramatically reduce ASO toxicity. The question is, so can we generalize this observation using even large number of ASOs? So for this purpose, we selected more than 300 ASOs. Some of them, based on the historical data, were toxic ASOs, and also majority are unpaired screening, like the normal screening for the lead oligo selection. What we found here, based on the in vitro study, without O-methyl at position 2, about 53% of oligos appear to be safe. However, when we introduce O-methyl at position 2, the safe ASO number turn to more than 80%. So this is more obvious in this figure. Here the x-axis is the log2 ratio between the parental ASO compared with the mock control. So towards the right is the more toxic ASOs. The y-axis is the log2 ratio between the 2'O modified ASO compared with the parental ASO. Towards the bottom, it's more safer. Clearly, there's a strong trend, OMAZO at position 2 dramatically reduce toxicity. So based on the statistics, we found more than 90% of toxic ASOs could be improved in this way. So in a more detailed dose response study in vivo, we found that introducing OMAZO at position 2 dramatically increased the therapeutic index as compared with the parental ASOs. We found with more than 30 pairs of ASOs in vivo, and also improved the therapeutic index was found in different tissues, such as the liver, kidney, and in different species from mouse, rat, and monkey. And also, it can reduce toxicity even in longer-term studies. I may not have time to go through the more detailed mechanism we proposed, but in short, toxic ASOs can bind more protein more tightly, which can cause protein mislocalization to the nuclear line in an RSH1-dependent manner. This can cause protein degradation, some nuclear protein clearance, followed by nuclear stress and apoptosis. When we introduce OMAZO at position 2, we can reduce protein binding and also reduce toxicities, so leading to improved therapeutic index. I hope I have convinced you that the toxic ASOs can bind more protein more tightly, and it can cause rapid protein mislocalization, degradation, and nuclear stress, followed by apoptosis. And introducing OMAZO at position 2 in the gap dramatically reduce protein binding, mislocalization, toxicity, leading to improved therapeutic index for majority of the toxic ASOs. So if you have questions, I will be glad to take, or I will invite you to look at the paper which was posted at OTS website. Thank you.
Thank you. Thank you, Liang. Are there questions for Liang? Yes?
Sorry, so there are a lot of toxicities associated with phosphorothioate oligonucleotides.
Right.
There were two distinct flavors of hepatotoxicity, a rapid onset and a long onset.
Yeah.
And then there's renal toxicity, and then there's thrombocytopenia.
Yeah.
So have you addressed both of the flavors of hepatotoxicity with this 2-O-methyl modification?
Yeah. The first question, after first one, we mainly focus on the liver toxicities, but it's also beneficial for the kidney. In terms of the acute and accumulative, for example, toxicities, we do find some ASOs that show toxicity with the late onset.
Right.
It can also improve in this case.
And then with respect to renal, there's
Yeah.
there's renal that's proximal tubular, there's
Yeah.
renal that's glomerular. So for
Yeah.
example, 104838 appears to be glomerular. Have you addressed that?
Yeah, we just did a very brief early study. It did improve the renal toxicity, but probably for a subset of ASOs. Maybe there are some other additional mechanisms. For example, we still have some outliers. For 10% of toxic ASO, we cannot easily fix by the position 2 OMAZO. Yeah.
Okay. Question there.
Yeah.
Thank you for a nice talk. So,
Welcome.
so it seems like you used like a 30-10-3 design for your study. So by adding like a 2' methoxy modification at 2 position, so from somewhat different perspective, your modification like become like a 5-8-3 design. So if you compare the general toxicity of ASOs having like 8 DNA at gap region and 10 DNA at gap region, do you see better safety profile of the ASO having 8
Yeah.
DNA gap?
That's a good question. I don't have a great answer for you, but based on our study, it's not due to the shortening of the DNA gap. For example, if you put OMAZO at position 9, which also have a 8 DNA gap, and you didn't improve the performance. With the 383 compared with 310-3, sometimes it's difficult to compare because you have different lengths, they have quite different binding profile and TM. There are too many variables there.
Okay, so there's 2 more questions that I'm going to allow. So each of you, 1 question each, and short question and short answer, please.
Okay.
Well, thank you. Great work and congratulations for your
Thank you.
paper award. I'm just curious to know whether have you used a charge neutral backbone like PNA or PMO for their behavior, the paraspecific protein localization?
Short answer is yes, we did. We used the MOP, the neutral backbone modification. We also observe the same positional effects. At position 2 or 3, we dramatically reduce toxicity with similar activities. So this was published last year in NAR. It was selected as a breakthrough paper as well. So you can look at that paper. We also look at actually does OH, 2'OH, or RNA positional walk. It also appears at position 2, you can dramatically reduce toxicity because it's less stable. We didn't go further with that. Yeah.
Okay, last question.
Thanks for the talk. I was just wondering, did you look into if you can improve the therapeutic index of already safe oligos?
Yeah, that's a great question. If it's already safe, I don't know why we want to do that.
Just wondering.
Nice short answer.
Yeah.
Good answer. Good.
Okay, with that.
Okay. Yeah. Thank you. Yeah. Thank you. Yeah. Okay. Thanks.